Welcome to the SBGrid YouTube channel. Software tutorials by developers, lectures by structural biologists, unique content brought to you by SBGrid.
So my name is Nicolas, and as you've well introduced, I'm going to talk to you about various methods to deal with thicker samples than what Liza talked about, which is more than 100 nanometers in thickness. So I got my PhD in France, and I worked on the ultrastructural characterization of plasmodesmata, which are small nanopores that connect plant cells to one another. And my work heavily relied on plastic sectioning of chemically fixed samples and also high pressure frozen samples. And so I did a lot of tomography on plastic sections. And so my main findings were to uncover a morphological maturation process going on in plant tissues of these nanopores called plasmodesmata. And so after my PhD, I wanted to continue electron microscopy because that's probably the only thing I really like in science. Technically speaking, I mean, and so I decided to step it up a little bit, and I wanted to get experience in cryo-electron tomography. So I came to California to work in the Jensen and the Meyerowitz lab, and now I work on the characterization of the plant cell wall by cryo-electron tomography. We're going to go through the different ways we can deal with thick samples. So this is kind of how I think about things when I'm interested in a tissue. So I have a sample. Here it's the sample of your dreams. And there's two questions you need to ask yourself is how you can preserve it. And when I mean preserve it is without inducing too many artifacts that are generated during the preservation steps, and then how to make it thin enough for transmission electron microscopy. So there's a whole part of sample prep that relies on room temperature chemical fixation, and we won't talk about this. We'll focus on the cryo-electron microscopy. So by how to preserve it, it means how to achieve successful vitrification of our sample. Liza very well explained, Liza very well explained the principles and what you should care about when you're plunge freezing. And usually samples that can be plunge frozen can be directly observed by cryo-electron microscopy. So it's a two-step process. But unfortunately, when we're interested in samples that are above a certain range, we need to resort to thinning methods. We'll be talking a lot about transmission electron tomography, so I just want to have a very quick word on it. So what exactly does tomography entail? It means that we have our specimen here in panel A, where it says ice, and this specimen is tilted at various degrees, and at each of these tilt angles, we acquire a 2D projection. And then with the collection of these 2D projections, we can come back to the 3D volume. So cryo-electron tomography allows to reconstruct the 3D volumes of our specimen of interest. Liza touched about single particle CryoEM, mainly that deals with samples that are approximately 100 nanometers thick. Wholesale cryo-electron tomography, meaning I can just plunge freeze my cell and stick it under the CryoEM. That can be done with cells that are at a maximum 500 nanometers in thickness. And when I say 500 nanometers, I'm stretching it a little bit. It's usually less if you want to get good quality tomograms. So what do we do when we have samples that are thicker than 500 nanometers in thickness, which basically means every sample, every cell, except a few bacteria? Well, we need to resort to thinning methods. And so we're going to be talking about two thinning methods that require cryo-focused ion beam. We're going to talk about cryo-focused ion beam on plunge frozen samples, and also this new variant called the waffle method. And I'll also just very rapidly talk about two others, because I think it's good that you also know that there are more exotic ways of thinning samples that are even thicker. And so basically, cryo-FIB milling can be performed on plunge frozen samples up to 10 microns, which is the limit of what can be vitrified at ambient pressure. And then anything that's above 10 microns will need to high pressure freeze it. So here the first case is my sample is thin enough for plunge freezing, but it's not for direct observation. So we're observing something that's in the ranges of a few microns in thickness. So the general workflow here is that we are going to plunge freeze our sample the same way Lisa described it. We're going to thin our sample by using cryo-focused ion beam milling, and then we're going to transfer this grid in the Cryo-TEM, and we'll acquire data on the thinned lamellae. So what exactly is FIB milling? Well, FIB milling takes place in a scanning electron microscope that is mounted with two beams, two guns, the SEM guns, that's the regular electron gun, and the FIB gun, which is a gallium ion beam gun. And so these focused gallium ions, they are very powerful, and they can erode the sample really fast. And so the game here is to take a sample that's too thick and to focus the gallium ions in rectangle shapes, and we erode away the sample, and all we leave is a really thin lamella that you can see here in panel 3. And this thin lamella is usually in the range of 200 nanometers in thickness, and that's amenable for cryo-ET observation. And so then these lamellae can be observed by CryoEM. And so here I show a lamella of a pollen tube, and you can see that when we erode away above and below the lamella, we're left with a thin vitreous sample where we can do tomography. And here we can see all the insides of the pollen tube in a near native state. And here I show examples of tomograms acquired in such lamellae, where we can see all the different organelles that are in the cell. And so FIB milling gives access to the inside of cells that are usually too thick for direct observation. So now, a little bit more difficult, we have a sample that is too thick for plunge freezing, and therefore too thick also for direct observation. So in these cases, we need to do something called high pressure freezing. So first, I want to very quickly go over what exactly do we need to achieve when we're trying to vitrify our sample. When we're vitrifying our sample, we need to freeze it with a high enough freezing rate so that we reach the glass transition temperature without leaving the time to the water molecules to rearrange into a crystal. And so this can be done on very thin samples at ambient pressure, which is why it's applicable on thin cells and thin and small proteins. But when we want to do it on thicker samples, we need to increase the pressure. So by increasing the pressure, what happens is that we inhibit crystallization. And so by doing this, we lower the freezing point and the nucleation point of water, and this reduces the rate of crystal formation. And so that allows to freeze or to vitrify samples that are below or equal to 200 microns in thickness. I'll pass this. And so once we've high pressure frozen our tissue that is 200 microns, for example, it's still way too thick for FIB milling and for observation. So here I'm going to show a variant of cryo-FIB milling called the waffle method. So the waffle method was developed at the In Situ by the Alex Noble team in New York. And so what they do is they take a grid and they high pressure the grid with the sample on it that is squished on the grid. And so that's why they call it a waffle, because it kind of looks like a waffle. And then you can add a cryocorrelation step where we screen the sample by a fluorescent microscopy. So here, those are plant cells that were processed through the waffle method. And here we're imaging the autofluorescence of plant cells. So basically the plant cells are everywhere. So we don't really care about like FIB milling at specific points. And then the FIB milling occurs in two steps. So here we're dealing with samples that are much thicker in the range of 20 microns. And so the waffle milling has a first milling step called the trench milling. So here in A, you can see the waffle. In B, you can see the trench milling. So when we trench mill, we present our grid, which is here, this orange stick. We present it orthogonal to the FIB gun. And so we mill directly straight into the sample to clear out all the thick material that is around this wedge right here. And then we tilt the grid at a specific angle, and we proceed at doing regular FIB milling on this little block that was previously cleared out. And so by doing this, we can generate lamellae on thick vitrified samples. And so here are examples of two waffle lamellae that were done on plant cells. And this allows to observe them by cryo-electron tomography afterwards. And so here you can see a snapshot. This is just a 2D projection where we can see the cell wall. You see all the fibers, all the cellulose fibers in the cell wall, the cytosol here, and here you have the vacuole. So why this waffle method can be not only interesting to weird people like me that do tomography on plant samples, but they can also be interesting for structural biologists. Since we're high pressure freezing our sample, we don't have any air in our system, so we're not worried about any air-water interface. And so the idea would be to generate waffle lamellae on high pressure frozen solutions of your protein of interest. And then a single particle can be either performed on the lamella, or tomography can be performed on these lamellae containing the little proteins. Just to finish, additional thinning techniques. So one that's quite old now, but it's getting replaced by FIB milling, but it can still be very efficient if sufficient expertise is available in your institute. It's called cryosectioning. So here we high pressure freeze the sample and we cut frozen hydrated sections that we lay on grids. And then we perform the tomography on these frozen hydrated sections. Another kind of exotic technique was to do cryo-FIB milling into a high pressure frozen sample that was embedded into a cryoprotectant that could sublimate at cryogenic temperatures. And so when we sublimate this cryoprotectant, we make, here it's a C. elegans worm. It is made available for the FIB milling. And the one that maybe you've heard about called the lift out, which is a really fancy way of FIB milling into really thick samples, and with like nanoscopic tweezers grab the lamella and then transfer it to another grid and perform the final milling steps. And then it's possible to observe these lamellae under the CryoEM. I've included a lot of interesting references if people are interested in these methods. And yeah, with this, I'd like to thank you for your attention.
Very nice, really ultra cool. I'm sure there'll be a bunch of questions, William, and people are availing themselves now of the Q&A tab at the bottom of the screen, and they'll type in questions, and then we'll get to those at the end. So thank you very much. Let's move on now to our third speaker of the day, who's Sarah Weaver, who will be talking to us about micro-ED sample preparation. Thank you very much, Sarah. We're ready for you to begin.